I'd like to start by thanking the organisers for this opportunity to present our work on RyR calcium leak triggering fibre calcium redistribution. In the muscle fibre, we have contractile proteins, and each myofibril is surrounded by sarcoplasmic reticulum, and the transverse tubule, which is an invagination of the plasma membrane. If we look at these junctional membranes, large version, we can see the T system, which has voltage sensor on it, which senses the action potentials to trigger calcium release from the RyR receptor on the SR membrane. And the other very interesting proteins relevant to this talk are STIM1, which senses calcium depletions inside near the SR membrane to trigger calcium influx immediately from the T system in response to the RyR receptor activity. So we're really interested in what's happening with the RyR receptor, how it's controlling all these movements of calcium. And a mouse that we're able to use, which has been published by Phil Hopkins and Paul Allen's lab of late, is this variant of the RyR receptor, which mimics MH susceptibility in humans. Work has been done on intact cells to show in these mice there's a gene dose effect. As we get a single RyR mutation, we get an increase in resting cytoplasmic calcium. There's two mutations. There's further increase in resting cytoplasmic calcium. Of course, the mouse is susceptible to MH, so it responds to volatile anesthetics such as halothane. Here at low concentrations, you can see that resting calcium-dependent fluorescence increasing in gene dose-dependent fashion, and further in response to higher concentrations of halothane. So what's interesting here is that in these heterozygous mice, and of course in humans with the single gain of function RyR mutation, there's no phenotype in the absence of a trigger. So that we suspect there's changes in calcium in the muscle in this condition, in the calcium handling, and also that males are more susceptible than females to dying under these conditions when they carry these mutations, which hasn't been explained. So our aim was to determine the effect of RyR calcium leak on calcium handling in the resting muscle and the effect of these changes on the function of the muscle. And I'll briefly go over the technique used, which is mostly mechanically skinned fibres, and we want to image calcium in the T system to measure leak and measure calcium handling properties of the T system membrane. So what we do is we bring a calcium sensitive dye close to the muscle fibre and it diffuses into the transverse tubular system, and then we can peel off the surface membrane, and that causes the transverse tubules to seal and trap the dye. And you can see here on a confocal image from a rat fibre treated in this manner. So on the next slide, I've got a schematic of the junctional membranes here to explain what we're doing. We have dye trapped in the T system, and what we expect when we have, of course, the SR next to it is at rest pumping calcium into the SR and leaking through the RyR receptor. We get an increase in local calcium concentration between the T system and the SR. In this region we call the junctional space. And this will cause the PMCA to work harder when calcium is increased due to RyR activity, and we'll be able to measure that change in the T system. And to see the actual effect of the RyR receptor, we can block the RyR receptor with tetracaine, for example. The calcium in the junctional space becomes the same level as that in the bulk cytoplasm, and therefore we expect the calcium in the T system to drop. Here's an example of that in a wild-type mouse fibre. This is the T system calcium transient. If we put on caffeine and deplete all calcium from the SR, we get rapidly activated store-dependent entry. If we wash the caffeine out and put on a standard solution, in this example 200 nanomolar calcium, we get a calcium transit that increases over 100 seconds, and we get to the steady state. After this, we can block the RyR receptor to see the effect of blocking the leak on T system calcium handling, and indeed we drop the steady state of the T system calcium. And that's our indicator of RyR leak. And if we do this experiment over a range of cytoplasmic calcium concentrations and across our genotypes, what we can see is this: a delta calcium T system, our indicator of leak, It increases as we have RyR mutation. The heterozygous mouse increases further in the homozygous mouse, and we find it's the greatest in a calsequestrin knockout mouse, which is also displays susceptibility to halothane and similar external stresses as the RyR knock-in mouse. We also, this data here is collected from male and female mice. If we separate them, we couldn't see a difference with this assay in the wild type. However, in the heterozygous, we could see that the males were leakier than the females. This was the case also in the homozygous and in the calsequestrin null. So there's a gene dose effect on leak, and there's an effect of sex, so showing that males have leaky RyR receptors, and also the absence of calsequestrin introduces a very leaky RyR receptor. So how does RyR calcium leak affect fibre calcium content? Well, we used a baptolysis technique that was introduced by Fryer and Stevenson in 1996. It's a skin fibre technique where the membranes are lysed and the fibre is surrounded by oil, which traps the released calcium there to bind to the contractile apparatus and generate force, which is used to calibrate the amount of calcium that's been released from compartments holding calcium. Using this technique, we determine the SR calcium content versus the leak. And what we could see is the wild types, of course, had the highest SR calcium content. Indeed, the female had more calcium than the male, indicating consistent with the males being leakier than the females. And this relationship of SR calcium content increasingly was a negative one, but in each case with the genotypes, the females had more higher SR content than the males. Calsequestrin null mice, of course, had a departure from this relationship because they had lower SR calcium buffering. So what does this mean? So we know that in the intact cells, it's been measured the increase in cytoplasmic calcium. There's a decrease, we've shown a decrease in the SR calcium concentration. So some might presume leaky RyR receptor scores increase in cytoplasmic calcium. However, cytoplasmic calcium must equilibrate across the T system membrane. So if you increase the leak, calcium must be pumped out in this direction. And if there's just an acute leak, calcium will be pumped out till it equilibrates again. So, but we need to explain this increased cytoplasmic calcium. We have measured across these mutant genotypes, steady state T system calcium was maintained. And if that's the case, that means this increase in cytoplasmic calcium must be due to an increase in PMCA activity to maintain the steady state, and there also must be an increase in leak to account for this increase in cytoplasmic free calcium. So we explored this further to indeed show it was the case by tracking PMCA activity. And here's the same experiment I showed you to detect leak, but in this case we use the T system calcium transient. We derive the flux rate. In this case, it's the wild type in the presence of 200 nanomolar calcium. And if we check that across a range of cytoplasmic free calciums, you can see here the peak T system uptake flux in millimolar per second. And we see in the heterozygous mice, this rate's increased. Homozygous mice increased further. And in the calsequestrin null, we see the highest rate of T system calcium uptake. If we take the Bmax from these the curves fitted to this data and plot it against leak, you can see we get what may be a linear relationship between the two. So we conclude that PMCA activity is increasing with RyR leak, and it means that the calcium influx is also increasing across the T system with RyR calcium leak. So the change in T system calcium permeability is what's responsible for the increase in resting cytoplasmic calcium in the presence of a leaky RyR receptor. And the next thing we wanted to do, because we expected that increase in cytoplasmic calcium would affect calcium in the mitochondria was to measure that. We advanced the baptolysis technique to be able to measure that total calcium in the mitochondria. And I'll go straight to the data. Wild type had the lowest amount of calcium in the mitochondria, consistent with having the lowest cytoplasmic free calcium. But we could see a sex difference as well. There's more calcium in the male than the female. In the heterozygous mice, there's an increase in total mitochondrial calcium, again, more in the male than the female. The increase again in the homozygous, more in the male than female, and a high amount of calcium in the calsequestrin male as well. We plotted these results against what we predicted to be the cytoplasmic free calcium from the published data to generate this relationship. And what's interesting here is as there's small change in the resting free calcium at this lower end, there's a steeper rise in mitochondrial total calcium. So what are the effects of calcium redistribution on force, and how does this redistribution look in the total fibre? Here is our results from in the SR, the mitochondria, and the cytoplasm. We also know what's in the T system. If we look at the wild type in the RyR knock-in mice, we get about the same total amount of calcium in the fibres. And what's changing is it's the distribution of calcium inside the cells. Of course, with leak, the SR calcium concentration decreases. This is going to activate store-dependent entry. And when we get store-dependent entry, the cytoplasmic calcium content increases. So it's not refilling the store because the SR remains leaky with a leaky RyR receptor, so it can't refill in these cases to the same cases as in the wild type. The increase in cytoplasmic calcium directly affects the mitochondria, and mitochondrial calcium increases. The other thing that happens is that in the calsequestrin mice, of course, they are capped with the amount of calcium they can hold in the SR, much less than in the homozygous, which has equivalent RyR leak and equivalent cytoplasmic calcium. We can see this, we get the same mitochondrial calcium in these fibres, indicating it's not the calcium in the SR that's determining that in the mitochondria. It's the cytoplasmic calcium that's determining the calcium in the mitochondria. So how do these results affect EC coupling? We measured twitch force recordings in whole EDL muscles. And what we found, of course, wild type here in black, our heterozygous mice in blue, we got basically the same responses, which we were expecting. We also know that the calsequestrin null, which has been recorded by Chris Ward's lab, there's no difference in twitch force in calsequestrin null and wild type. However, our homozygous mouse showed much less force than the wild type. Chris Barclay in my lab has developed a model for determining the calcium transient from using input properties of calcium in compartments, as well as all the other relevant parameters, and matching it to the experimental recordings that we've made, and that from Chris Ward's lab. And he was able to determine major differences in the amount of calcium released to generate the same force responses in our wild type head and calsequestrin null mouse. So the effect of this redistribution is the amount of calcium release, which is normally 300 micromolar with the first action potential in a train in wild type, is reduced to 260 micromolar in the heterozygous. And therefore, the amount of calcium in the cytoplasm is already compensating for the reduced amount of calcium in the SR. So we need to release less SR calcium to the cytoplasm to generate the same amount of force that we do under this situation, where at rest there's less cytoplasmic calcium in the resting fibre. In the calsequestrin null, we already have a lot more calcium in the cytoplasm than in the heterozygous. It releases less calcium to produce the same amount of force here, which is not shown because it's published by Chris Ward's lab. and therefore it needs to release even less than the heterozygotes. Our homozygous mouse wasn't able to produce the same amount of force. We calculated it released 130 micromolar calcium. This redistribution of calcium in this mouse was not enough to compensate for the 2 mutation in the RyR receptor, so we presume that the calcium release is directly affected by these mutations in this protein. So I'd like to move to the conclusions. When we have a leaky RyR receptor, we predict that we're generating nanogradients either side of the SR terminal cisternae membrane to control what's going on with the calcium distribution in the muscle. And this is incredibly important for the function of the muscle, and it's also very, very precisely controlled by this leak of the RyR receptor. So when the RyR receptor leaks, there's a depletion here behind the terminal cisternae membrane. There's direct evidence from this from Jenny Yang and Francesco Zorzato's lab recently. So when this becomes more leaky, we dissociate more calcium off STIM1 and we get chronic activation of store-dependent entry. And this is what's going to increase the resting cytoplasmic calcium. This will increase the rate of SERCA activity. More calcium is pumped into the SR. However, the SR doesn't get to the point of the amount of calcium that's held in the wild type as it does in the mutants, because we maintain a leaky RyR receptor. So we have this standing gradient. And the SR, even though one SOC is turned on, it is not able to refill the store. It's putting calcium into the cytoplasm. What's maintaining the T system calcium gradient? Well, we end up, the leak also increases calcium in this space. We predict that this higher calcium is binding to calmodulin to bind to PMCA to change the affinity of calcium for PMCA, and we pump calcium out at a higher rate. Robin Murphy did Western blotting for us of these genotypes, and we couldn't detect any difference in PMCA density across the mutants compared to the wild type. So we must conclude that there's a change in calcium affinity of the PMCA under increased leak. So the consequences of these changes cause pre-saturation of cytoplasmic binding sites in the resting muscle to relieve calcium release needed for force generation. Increased cytoplasmic calcium will push more calcium into the mitochondria, and higher free calcium in the mitochondria increases the rate that ATP can be synthesized. And this is certainly would be important even under the basal condition where the SERCA needs to work harder in these mutants. Therefore, I'd like to finish by thanking you for listening, and also thank the guys in the lab who did all the work. Luke Pearce did all the T system confocal stuff. Cedric Omblay did the baptilysis technique to show you the SR and mitochondrial total calcium content. Crystal did measures of free calcium in the mitochondria, which unfortunately I didn't have time to show you. And others in the lab certainly were involved in the experiments, Daniel Singh and Aldo. I'd like to thank our colleagues, also Chris Barclay, who did the modelling, Robin Murphy, Chris Vanderpoel at La Trobe, Phil Hopkins, Paul Allen Vickers, and Robert Parton and Harriet Lowe, also at the University of Queensland. Thank you for listening.